All right, thank you. Really appreciate the opportunity to be here today and to tell our story of the innovative ways in which we're applying AI machine learning. It was really insightful to hear all the presenters speak today, to attend all the talks yesterday, to have conversations amongst all of you here, and to hear all of the impactful applications of AI, those really transformative ones, especially, those that where AI is enabling processes that we weren't doing before. Those are really the ones that I find, exciting and love being part of. In fact, I was out at dinner last night, and a few of the other fellow speakers and many of the other people that I've chatted with here today so far seem to have also started their AI journeys through the analysis of scientific data, through high-dimensional data sets in physics and chemistry, and life science. And I'm kind of curious here today, just by show of hands, how many people here have that background in scientific research? We got a few? All right. What about life science specifically? That's pretty good. There's a lot of people here that, that will get a lot of that deep content here as well. So, disclaimers aside, here's a secret. Here's a secret about drug discovery. In drug discovery, everything is a model. We talk about a cellular model, a disease model, a structure-activity model, an animal model. All of these are scientific models. The pharmacokinetic models that determine if a model will be sufficiently, if a molecule will be sufficiently distributed when absorbed by the body. The pharmacodynamic models to determine if the drug will deliver the therapeutic effect of interest. These are all scientific models, each one being a proxy built by expert scientists over decades that, when used correctly, will inform on that one ultimate grand experiment at the end of the tunnel, the clinical trial. And as time goes on, our industry has accrued more and more of these data points, molecule by molecule, program by program, decade by decade. Data sets turn into databases until eventually data scientists start analyzing the trends, finding patterns, performing searches, simulations, clustering, visualizations, until ultimately, in AI-based drug discovery, we model, ML, all the models scientific, right? We build machine learning models to represent all the experiments that we've collected and accrued. Each data set gets its very own model, models that must grow and be retrained and be versioned and be stored and accessed by many of the groups that are running different projects. This, of course, is why we need those modern AI stacks, right? With robust ML infrastructure practices, version controls, modern model serving capabilities. But again, now what, right? We could use these models to carve back on a proportion of the experiments that we might have otherwise run. We can enrich our programs for better outcomes, perhaps run more programs within those domains of applicability that we've set out for each of the models in the first place. But in a sense, we're not so much challenging how drug discovery happens, so much as we're augmenting or finding efficiencies. Moreover, if all of this data is really segmented across different companies, designed around their own core competencies and their own therapeutic areas. And really, this is the story of biotech. This has been the story of AI-based drug discovery so far. Finding all these scientific models, finding all these data sets, and developing machine learning models to represent them. I'm here today to tell a different story. Those applications that really excite me are the ones where you take different forms of data and you use them to transform and to create predictions for things that you weren't otherwise predicting before, right? Those in which you're using machine learning to enable new applications, to enable the types of insights that you weren't getting before, to redesign how data gets collected, what kind of data you can do, and how you can scale that data collection. So Recursion's story is that of scalable, high-dimensional data. robotic systems to industrialize data generation and data collection. Data, people, and compute. So, rewinding a little bit, when we try to understand biology through the lens of an individual program, one at a time, through an individual target of interest, an individual pathway, we're casting this very narrow light on that landscape of opportunities. We're roughly charting out biology one pathway at a time, charting out the coast of an island in this complex biological landscape. We're also, and when we start programs with AI, we get drawn to these lights. We get, like these are the lights, the beacons, where there is known biology that has been de-risked, that there is a lot of insight, that there is a lot of data. Things that have been seen and targeted by others, following in the footsteps of past explorers. But instead, as we're industrializing our experiments and our data collection, that landscape looks quite different, right? Rather than looking at everything on a case-by-case basis, we can take this bird's eye view of all the opportunities and can instead choose those opportunities that look the most promising. Those that fall under new regions, creating medicines for populations with an unmet need. So just to make this a little bit more concrete, let's go through that first form of data collection. Let's start with cells. Specifically cell images. These are information-rich and inexpensive. These are the cornerstone example for the high-dimensional approaches for drug discovery data here at Recursion. All of these are microscope images of cells. So we got two different systems. On the upper left side, we see, of each system, we see a disease state of a cell. On the upper right side, we see the control, how the cell should look in a healthy state. And the goal here, and the overarching goal of drug discovery, is to find a new compound, a new medicine, that when you start dosing the cells, when you start dosing people, at increasing concentrations, you start seeing that disease state get restored and start seeing these look healthier and healthier, right? And the main point here is that these images are this information-rich and inexpensive to generate. And they're scalable. Right? In fact, we run these experiments on full plates with 1536 wells, where each well is one experiment. We add cells into the well, and we perturb those wells in different ways. We add compounds to those wells. We knock out or we remove genes from the cells one by one across different wells. Each one representing modeling a different disease or a different treatment. So this is not unlike the microscopy images that the oncologists, the pathologists, and the research scientists do in the lab, that they use to diagnose conditions or suggest therapies. But unfortunately, we can't scale those oncologists. We need a better way to read those images. Instead, our images are analyzed by masked autoencoders, a self-supervised approach to machine learning, in which the goal for our models here is to do image reconstruction. So we train masked autoencoders on this image reconstruction task, such that they can reliably reconstruct images starting with only a randomized 25% subset of the initial image. We've also shown that it scales with the architecture and the amount of data on hand. Last June, we recently upgraded our on-prem supercluster with over 500 NVIDIA H100s, and quickly wanted to test out these new GPUs. So the first thing that we did is we increased the size of these models. Our original version of this masked autoencoder had 300 million parameter model. We upgraded it to a 1.8 billion parameter model. But rewinding there for a second, why would we do that? Why would we do, the goal here isn't to generate images of cells, right? Why do we get that question quite a bit. Rather, if the model is able to reconstruct the original image, it's able to interpret what's happening inside the cell, right? To notice those morphological abnormalities, to notice when something is elongated or a part of the cell is larger than it should be or smaller than it should be. Those intermediate layers between the encoder and the decoder actually carry a lot of that really meaningful information that could be used for downstream tasks. These can be used to determine if images are similar to one another or different from one another, and how they differ in which ways. You could tell if a cell perturbed by a compound or had its gene removed are sufficiently different from our controlled or our unperturbed cells. And what we found is that our new 1.8 billion parameter model is now able to detect 20 to 25% more of the genes that we expected to see based off of our benchmarking data sets. And this opportunity space is huge, right? 25% of the genes is more things that, more regions of biology that we can now see. It's more, like it's a bigger light that we can kind of cast on that map. And more therapeutic opportunities, more medicines we can discover. But there's also a problem. To date, we've run over 180 million experiments. Since the last time we did this exercise of upgrading our models, we've steadily increased the number of plates that we've analyzed, maybe doubled the amount of data that we had. We've scaled up our model by 6-fold, right? All of these homegrown systems that we had to run inference on it, they were starting to reach their limits. We couldn't necessarily get the observability on it. It would take months for us to run these on. And that's really where our journey with AnyScale comes in. So we're pretty new to the AnyScale family. We've been part of our tech stack now for about 3 months. And we, like last week, the last couple of weeks, we really put that here to the test, right? So we called up our cloud compute provider, asked them for 4,500 GPUs. This was a new model architecture and a new GPU type that we hadn't dealt with before. And we wanted that data. We wanted that data fast. So this was kind of our first real application of that AnyScale system. We were able to get that going. Very few issues. Got all the images inferenced in a day. And one of the really meaningful things here is that we didn't have to spend as much of the time figuring out that inference stuff, figuring out the compute, and figuring out how we would distribute all of that data. Instead, our data scientists are able to spend their time asking, how do we properly align these embeddings? How do we aggregate them? And how do we transform them into useful representations of biology? So these embeddings allow us to relate experiments. That's really the value of the machine learning, of the AI in this case. It's what allows us to compare these images. So here in this plot, every dot that you see is a different gene that's been removed from the cell, and how similar those cells are to one another with that gene removed, right? So it's colored by different groups of genes that when removed give a very similar effect on the cell. And that means they're related functionally. It means that when the cell functions, the molecular pathways that gene is involved with are impacted in the same way. And these embeddings also allow us to project all of the compounds that we have in our library. Right? We've got these corresponding experiments where instead of perturbing the cells with gene knockouts, we perturb them with compounds. And we can see that the embeddings for those compounds look very similar to certain genes, implying that they mimic those genes, that they impact the same areas of biology as those genes. And really, this is the landscape from which we can mine those new opportunities, that we can kickstart new programs. So stated a little bit differently in tech bio, right? Where we discover models of biology. And in this context, we're no longer talking about models of models, we're talking about new models, right? Like we're intertwining kind of the concept of the data with the concept of the machine learning. Data that standing alone isn't as useful, and vice versa with the machine learning, right? Machine learning is what's providing us with that relatable representation across the different facets of drug discovery. It's what allows us to build new scientific models as well as ML models by revisiting the nature of the problem itself. So connecting across these layers is what allows us to fuse the strengths and limitations of each of these modalities, to couple the scalable representations, like the cell images and the phenomics, which those that instead provide more detail, that might not be as scalable, to tell us which routes to pursue and which ones lead to dead ends. So to recap on our phenomics, these are our cell models. We continue to scale and push the dimensionality on multiple fronts, adding more compute and innovating on the assay itself. What you see here is actually the next version of the assay, where instead of staining, like fixing the cells to the plate and staining them so that we can really examine, so that the experts can look at the cells and see what's happening in the various subcellular localizations, instead it's bright field imaging. And these are, while they're less interpretable to humans, the AI models extract just about as much information from these images as they did with the stained images. And that provides an opportunity, an operational advantage in the lab for them to create even more plates. The other opportunity that it provides is because we're no longer fixing these to the wells, we can take snapshots at different points in time, and we can start adding another dimension to the problem, time. All right? So the really interesting thing here is that we're starting to trade off human readable representations for those and start optimizing them for the AI instead. So another data layer here is the transcriptomics, right? This is where we do gene sequencing, specifically in the very same context as the cells in terms of removing genes or adding compounds. We do the same thing, but instead of taking photos of the cells, we sequence their transcripts. We can tell which genes get upregulated or downregulated in response to that compound, in response for that gene knockout. We can also do this at scale. Now, not the same scale as the hundreds of millions of experiments that we do with the phenomics, but still a substantial scale nonetheless, 1 million samples sequenced so far. Each of these samples with thousands of genes themselves, right? If I were to rewind 20 years ago, I spent a summer in a research lab, and I spent 4 months sequencing one gene. One gene. This is literally billions of times larger than that. Another data layer as well is we automate our ADME data collection. That is the set of experiments conducted to profile if a compound will be properly absorbed and metabolized in patients. These, of course, are supported by large chemistry models that excel at representing small molecule drugs. These robotic systems are able to profile about 750 compounds a week, and by using our models, like at this scale, even though it's significantly larger than what we can see in more traditional labs, it still doesn't reach the millions of compounds that we have access to, or the infinite or near infinite amount of compounds that we can design, right? So it's important for us to have these models to be able to see which direction we should be going. So every time we run one of these experiments, every week, when we get 750 new compounds in, we automate that model rebuilding and deployment process. And that was another system that we developed through the use of AnyScale over the last few months. And this is what allows us to record 750 molecule data points, but have that be applicable to thousands of molecules instead. Lastly, at the animal levels as well, here AI helps us detect and monitor mice in our digital vivariums. So these cages are designed to continuously monitor mice as our ML models determine if there are changes in their eating or sleeping or behavior patterns, to see if they're getting drowsy, right? As the mice are exposed to these new compounds. It's what allows us to front load safety in our programs. Lastly, we also develop LLMs to interrogate the data and to launch new analyses, to run models as well, right? But one of the key pieces here to highlight is that our model serving needs are getting much more complex. Our model systems are now starting to interact with one another. Our research labs at Valence are pushing the boundaries of generative design and multimodal systems, each with complex architectures requiring rapid inference and model-to-model interactions. So, and really, As we see more and more predictive models in complex ML systems, we're making this choice now to invest in that solid stack of ML infrastructure to accelerate development and enable future applications. And ultimately for what? Industrialization, the use of AI is what allows us to explore a much broader landscape of therapeutic opportunities, triaging those that will drive the largest impacts and address unmet needs. It front loads failure, as well as driving down the costs and the timelines. So the take home here is that really for us, AI is the means by which we consume the data. It's the unlock that lets us design scalable, high-dimension experiments that serve multiple purposes. We don't necessarily conduct an experiment at the time that the program is generated. We pre-run a bunch of experiments in order to identify what that landscape of opportunities looks like. It allows us to be more selective and more intentional with the opportunities that we act on. It allows us to simultaneously reimagine and intertwine both the scientific and ML concepts of what a model is, and what model should be guiding the future of drug discovery. Again, really appreciate the opportunity to speak here today, and I'm around all day for questions. Thanks.